Hello everyone, this is our series on science and technology. My name is Kshitij Dhiman and I'll be taking your lectures. So before starting, I would like to discuss one very famous quotation by Rudyard Kipling. He said that I keep 6 honest serving men. They taught me all I knew. Their names were what and where and when and how and why and who. So whenever you are studying about science and technology, and whenever you are studying about any subject in UPSC or any topic, be it a static part or current affairs, please try to remember these 6 words whenever you are making your notes. This will help you remember better and this will help you understand your subject matter in a better manner. Right? So with this framework, let's proceed. So in this lecture, we are going to start the topic biotechnology. The same approach which I mentioned in the previous quotation, I will be taking that. I will be discussing what is biotech or what are the implications, what are the techniques used and so on. So let's get started. By the way, this is first lecture in many series of biotechnology. We'll be dealing with biotechnology in a comprehensive way and similarly with other topics. So first of all, you need to understand what biotechnology really is. Is it an old technology or is it a new technology? One of the simplest examples that I can give you for biotechnology is the curd, dahi. That is also a product of biotechnology. Beer, making of beer, the alcoholic drink, that is also the product of biotechnology. Now, this will tell you that biotechnology is not something which has just presented itself in last 100 years. It's an ancient technology. All right. So basically, if you want to understand biotechnology, whenever there is a word in technology, whenever there is this word technology, that means the science that is being studied is being turned into a product that can be used for human consumption. For example, science and technology. We are studying science and technology. So we are studying science part also, and we will be studying the technology part. That is the implementation part. UPSC is interested in the application part. It wants to know if you know the applied part of the science. Nowadays, questions are not coming from the static part, like they are not asking basically about the digestion system of human body. Rather, they will ask something regarding to Internet of Things and so on, which they have already done. So we need to focus on the application part. So biotechnology is basically in which you use biological processes to create something that can be used for human consumption, basically a product. All right. One of the most famous techniques to do this is genetic engineering. Now don't worry, we'll be studying about genetic engineering shortly. Before moving on to the techniques of biotechnology, I'll be teaching you all the related keywords which are relevant. Now in biotechnology, we play with, let's say, DNA. We play with DNA, we try to change it so that we can either cure something or we can improve something. So, just a second. So we use DNA manipulation to create something, or let's say cure something or modify something. So we can change the DNA and do all these things. One of the most prominent examples of modern biotechnology is the insulin. The insulin that diabetic patients are taking nowadays, they are produced because of genetic engineering or basically biotechnology, in which it is created artificially. All right. Otherwise, obviously, no natural source can produce so much insulin that so many patients can take it. Also, there are some human growth hormones and sweeteners which are created with the help of biotechnology. Now, Carl Erke was the person who actually defined biotechnology. He said that all the lines of work just a second, by which products are produced from raw materials with the aid of living organisms. So this is very important. You need to focus on this term living organisms. Because as we will see in the further slides, in BT cotton, there is a variant of cotton, that is BT cotton. Now this BT is the part which was inserted with the help of biotechnology and which made cotton more resilient to pests, and it was also approved to be planted in India, right? So as I was talking about, is it a recent technology? No, it is not. In ancient world also biotechnology was understood and used, especially for beer making and preserving some things like pickle making, and as I told you, curd, dahi, right? So in ancient biotechnology, food, shelter, domestication, in all these things biotechnology was used some or the other form. Now fermentation, right? We use fermented foods like for example idli, it is prepared by fermentation. So that leads to basically understanding that biotechnology is not a recent phenomena. In modern technology, during 1960s and 1970s, people started actually using biotechnology with the help of machines or the modern machines. Earlier, it was more based on the natural process of things. Now, we have come into a phase in which technology is used to change the genetic things of any organism. So that is what is modern biotechnology. That means in ancient and classical biotechnology, more of the natural processes were used. In the modern biotechnology, more of the, let's say, technology is used, all right, not the natural processes, because of which there are many inventions that have basically brought to the brought to the fore. So here you can understand it little bit clearly that in biotechnology, this is the big circle, which is biotechnology. Now the modern biotechnology is part of this biotechnology circle. Natural regeneration of plants, cultivation methods in which farmers have been breeding different varieties naturally. Tissue culture, natural breeding. Animals also, there has been natural breeding of animals to get better and better genetic variants of, let's say, cow, which will produce more milk and so on. So modern biotechnology is the usage of modern technology which will lead to further changes like genetic engineering, in which we can change the DNA of a person, right, which has also resulted in genetic modified organisms. So this is the thing. You have to first understand that biotechnology is not a new thing. Modern biotechnology is the modern version of biotechnology. Let's now see some examples. From time immemorial, people have been breeding plants and animals. Herbal medicines were created, wine and beer, bread, septic systems, basically we can say sanitation systems, vaccines. One of the earliest application of biotech, modern biotech you can say, was to create vaccines. One of the earliest vaccines is smallpox, against smallpox, right? So biotechnology is something which we use in our daily life, which we see in our daily life. Alcohol, I have talked about beer, right? One of the most prominent examples. Then penicillin also. In all these things, to prepare all these things, biotechnology is used. Similarly, with the help of modern biotechnology, we can develop new medical treatments for many diseases which have been uncurable till now. For example, cystic fibrosis, right? Also, biotechnology has an application in environmental area also. For example, bioremediation. This you can study in your environmental section. So, what do you think? Is it a recent technology? I hope this question has been resolved till now. So, one question must be coming to mind, then why are we studying about it right now? Because as I told, in the 70s and 80s, lot of new technology came up, because of which we could do some things which were not possible before. For example, editing the DNA, editing the genes of one plant and inserting it into another plant. So these things have become modern biotech. Otherwise, we would not be studying about it. So we are studying because these things, these technology has just come up in the past 50, 60 years. Right? So the hype, or why we are studying it right now, is because of the new technology, new techniques, and of course science is progressing, so we need to know about it. As simple as that. Now let's see some definitions without which we cannot fully appreciate biotechnology. First of all, what is DNA? Although we have all studied about this, and some at least because till 10th we all have studied about this. So DNA is basically a molecule, right? It contains nucleotides. It contains, basically, rather than going into technical things, I would like to explain you its importance. So DNA is basically the map, or it's all your genetic map of how you are, of what you are, how organs will develop in your body, how much height will you gain, right? Will you be a basically athlete, or will you not? If you will be short person or a long heighted person, right? If you will have strong muscles or not. Many things depend upon genes, and genes are part of DNA. So it's basically an instruction of how to build you and maintain you. For example, if something happens to your cells, how to regenerate them, and so on. So it is all written in DNA. DNA is found in every human cell, all right, in nucleus. Every person's DNA is unique, but it is not unique if you have a twin, all right. So DNA is a structure, complex structure, which has the genetic information on how to maintain you and build you. So there are nucleotides, for example, namely guanine, cytosine, thymine, adenine. All right, these are also called, when they combine with each other, they are called base pairs. So base pairs are like ATGC, right? So this is how it looks in a double helix structure. All right. Now what is a gene? Now the part like previously you saw, this is the DNA. Now small parts of DNA is basically a gene. All right. If DNA like basically it's a paragraph. Gene is a paragraph in a DNA. Now what does this contain? Now like I told you, DNA contains the DNA contains the information on how to build you and maintain you. Now it's a big list. Now the small lists are genes. For example, how much height will you attain? That is one gene. Your skin complexion, another gene. Your hair quality, your nails quality, your athletic predisposition, everything like this is genes. Small aspects are genes. All right. So genes tells our cells to make molecules called proteins, and then basically human body develops. So DNA will not directly develop human body. It has instructions to create proteins, and those proteins make up the human body. For example, some genes tells the cells, human cells, produce this protein, and because of that protein, our body grows. Right? Now, when these instructions go wrong, or something goes wrong, that leads to something which I will tell you soon. Now, this is the way you can visualize. This is the DNA. Now, the small part is gene. Right? This part. So, focus on this small part. That is a gene. It is not the DNA. DNA is the longer part, gene is the smaller part, right? Now this is what I was talking about. If something like this goes wrong, like genes give instruction to cells to produce something. Now if any of the instruction goes wrong, because to create the human body, lot of cells have to replicate and lot of genetic information is there. If something goes wrong, then the cancer cells develop. So when a cell grows or basically divides, it has to create an identical copy of the DNA. Like I am a cell, I am creating another cell, right? Don't think of me as a human right now. Think of me as a cell. If I create another cell, so I have to give that cell an identical copy of the DNA. Now here mistakes can happen. This copying of DNA from one cell to another can create some issues which might lead to some small, error. Now, if that error happens, then the second cell will not produce proteins properly. And there is what cancer happens. All right. Now, this thing can happen genetically also. Some people are predisposed, basically genetic, what I mean to say is, such errors can be genetic also. All right. Other things that can damage the DNA are like tobacco, smoking, alcohol, which can create faulty genes. That is why you might have heard lung cancer, throat cancer of in people who smoke a lot or eat tobacco a lot. So this is how cancer develops. Now what is a chromosome? Now chromosome is actually the bigger part. It's like we were going down, right? DNA, small part of DNA. Now the chromosome, now going the opposite way. Now what comes above DNA? Rather, DNA combines into what? It combines into chromosome. Now to protect basically the DNA, it is put into a structure. Now you don't need to go into the specifics of it because obviously this is not a neat class, right? So we just need to know what happens. So to protect DNA, it is coiled into or wrapped up into a structure called chromosome. All right. Now how to imagine this? So gene was like a paragraph. Gene was like a paragraph, right? To create one part of you. So chromosome is like a chapter in that manual. All right. So DNA might be like a page. All right. Not exactly, but let's just compare these two, gene and chromosome. So basically there are 46 chromosomes, or rather 46 chapters in your manual. That is, let's say there is a book in which it is detailed how to create you. So in that there are 46 chapters which are basically 46 chromosomes. 23 comes from your mother, 23 comes from your father. All right. So why it is strengthened? Why DNA is strengthened? Because DNA strands are very important. They have to be secured and they are basically delicate also. So we need to package them very carefully. Like whenever you send a glass material through courier, you package it very carefully, right? So just imagine like that. So, all right. So this is how it looks. Let me zoom in for you. All right. So see, this is the DNA backbone, the black one, structure, helix structure. There are nucleotides in it. So then it is coiled and built into chromosome. This is chromosome. The X thing that you can see, it's a chromosome. Then it goes into a nucleus of a cell, and then it is like a human cell. All right. Okay. Another thing which we are required to know, what is a genome? Genome is basically everything. Everything means every DNA that you have, every detail of the DNA is genome. Simple. Whatever DNA you have is your genome. All DNA that you have, genome. Simple, very simple. So if DNA is a set of instructions that is organized into paragraphs, that is genes, then chapters, that is chromosome, then entire manual from start to end, the entire book is genome. All right. So data is DNA. So now let us summarize this. Genome is the whole book. Alright. The data in it is the DNA. It is organized into chapters and paragraphs. Chapter is chromosome, paragraph is genes. I hope now you understand. So all the genetic matter in a person is a genome. Alright. Let's move ahead. Now we come to the part where we see what is genome editing. This has been in news a lot. Genome editing is basically gene editing. It is also known as gene editing. So do not get confused. If you hear gene editing, it's the same thing. If you hear genome editing, it's the same thing. Now editing. Now how do you edit? Usually we edit by erasers, right? But it's a pencil. Now how do you, so it's a tool, right? So genome editing is also a technology or a tool that will give you the capability to change someone's DNA. Alright. So genome is basically all the DNA, as I mentioned in the last slide. So now you understand genome editing is basically DNA editing. So genome editing involves a lot of technology by which you can change someone's DNA. Now how will you change things? You can add something, you can remove something, or you can change something, right, in the genome. So this is how genome editing is, or basically explained. It's DNA editing, it's gene editing, it's genome editing. All three are same. Now how to do it? The technology actually that is used is called CRISPR-Cas9. This is the latest technology that is being used to perform this. And with these tools, we can actually save people's life. All right. Especially genetic things, because genetic diseases, many of them don't have any cure, because this is in your genes. It's like saying that you have created a building and then you go and change the foundation. You cannot do that, right? Building will fall. But if we can replace it by using modern technology, then why not? So this is the genetic editing or gene editing or CRISPR-Cas9. All these are new technologies which can help change someone's DNA or remove genetic diseases. So let's just briefly see what is like how it works. So CRISPR-Cas9 is like a scissor. All right. It basically a biological scissor, which and we humans got this idea from a bacteria which uses to cure something in itself. So we got that idea from a bacteria which uses this technique, not CRISPR-Cas9, but it uses a protein to change something in itself. So how it works? So first of all, a DNA cutting, just focus on what I am telling you, otherwise it will become very technical and which is also not required. So this is just for an information. UPSC will not go into this much depth. They have asked previously a question on this. We will see that soon. So they just ask what is CRISPR-Cas9. So I am just giving you for better understanding this thing, right? So let's see. Let's say this red part. Alright, so first of all, in a cell we insert a DNA cutting enzyme and a guide molecule. So DNA cutting enzyme will be the protein which cuts the DNA. Now here in the second picture you see the DNA, right? So this guide molecule will find the target DNA strand or the red part which we need to change. Guide molecule will find the red part, and then DNA cutting enzyme will remove that red part which you want to change. So this is the third image. In the fourth image, the DNA repairs itself with its own processes. So basically you have replaced the DNA. So understand, a cell, human cell is there, right? Or any cell is there, in that you insert a guide molecule and a DNA cutting enzyme. The guide molecule goes and find the thing which you need to cut, and the DNA cutting enzyme will cut it, and then it changes, right? So very simple process, that's all we need to know. Now CRISPR-Cas9, what does it mean? It means clustered regularly interspaced short palindromic repeats. So basically it will create something that will be identical. We don't need to go into depth of all this. Now the protein that helps to do this is protein 9, CRISPR associated protein 9, CAS9. So basically it acts as a scissor. This protein acts as a scissor, cuts the desirable part, and then DNA repairs like that. All right. So it's a genome editing technique that wherein you can cut a specific part of the DNA. So here with this help you can permanently modify the genes that are there in the living organisms, humans, animals, plants. With this you can remove the mutations which are harmful or which are creating diseases in your body. All right. Recently in China there is a team which has corrected, okay, they have changed the genetic mutation in 3 human embryos using CRISPR. All right. So basically they have changed the genetic information in embryos. All right. Embryos are very early stages of a human's birth. Next term which we need to know is recombinant DNA. Recombinant DNA. Basically, it's any DNA that you are constructing in the laboratory. Right? So it is a name given to process in which you take one piece of DNA and combine with another DNA. All right, and then you basically combine them. Hence the name recombinant. So you combine 2 or more different strands of DNA and hence create a new DNA. All right, and this is done mostly to combine DNA from 2 different organisms. All right. So, as I said here, recombinant DNA is a form of DNA constructed in the laboratory. This is the most crucial difference because the natural DNA is constructed in the body. So, where it is used, rDNA is used where? In the production of therapeutic molecules, insulin, hormones like growth hormones, use of genetic modification in environmental, or rather we can say the use of genetic modification. Okay, let's just take environment. Sorry for the grammar. Bioremediation. This is again something which you will study about in environment, but I'll just mention here. You use biological things like there are biological bacteria which can actually help in oil spills. So please do read about that. Then use of genetic modification agriculturally. All right. So here in can be cut. The use of genetic modification environmentally. The use of genetic modification agriculturally, like GMO. We'll study about GMO. So here rDNA can be used because what we can do is we can simply take one DNA of a bacteria, insert into another bacteria, and then make the second bacteria capable of doing something that the first bacteria was able to do. So just for an example, you take this bacteria, it can suck the oil, but it cannot live in water. Let's say for example. So you take a bacteria which can live in water, and you pick that, then it will have a DNA, right? Right. So the first one cannot survive in water, but can clear or eat an oil. Second one cannot eat the oil, but can survive in water. So what will you do? You combine both the properties from these different bacteria and create a new bacteria. Simple, right? So this is how you can create new things by rDNA. Now, what is DNA technology? Another term which I want to discuss is DNA technology. Basically, obviously, the name tells you anything related, any technology that is using DNA. All right. So, modern biotechnology. Again, I'm mentioning modern. So, modern biotechnology basically depends on DNA. We have to cut the DNA. We have to paste it somewhere else. We have to analyze the DNA. We have to see which part of the DNA we want to cut from some animal or some bacteria, like BT cotton. we cut the Bt part from another bacteria and then put it into cotton. It was heavily tested before releasing it to the public. Now, what is the example of DNA technology? Example is DNA cloning. Cloning, Dolly the sheep, right? So DNA cloning is basically you make copy, all right? Basically, if you want to create another sheep, right, or another human cloning, so you will create, you will take the DNA of the original person and create many copies of that DNA in another human, right? So that will become cloning. Polymerase chain reaction. Now, if I don't tell you, will you be able to get it? Because get it in the sense reference. So PCR, is anything shining in your mind? Are you reminded of anything? Now are you reminded? Just look at this term RT-PCR. So basically it's the COVID test. So DNA technology uses, an example of DNA technology is polymerase chain reaction, PCR. It's a DNA manipulation technique in which many copies of a DNA are created from a small part of DNA, right? So basically they put the swab into your throat and all, and then they collect the DNA, and then they create multiple copies of that DNA, and then they test it, right? Another example of PCR is forensic science. Let's say let's say there is a crime scene, and over there is just a drop of blood that is available. So they will take that drop of blood and use PCR to create more DNA out of that blood, right? DNA sequencing, this is very simple. It's basically they want to know what is the sequence of ATCG, the nucleotide basis in a DNA molecule. So very simple thing. With this, basically what happens is this is present in every human, but the combination or the sequencer is different. So they study and then they see how to change things, right? Another term which is related to gene editing or DNA technology is stem cells. This has been in news quite a lot of time, so it is very important for us to understand. Now see, our body is made up of a lot of cells, many different type of cells. Some cells perform special function like red blood cells. They carry oxygen around our bodies, right? But they cannot divide. Stem cells, they provide new cells as the body grows. Like a child is growing, you must be thinking that where do these cells come from? How does the bone grow? They grow because of stem cells, right? Also, whenever you're injured, like your skin regenerates, right? So it regenerates because of the stem cells only. So stem cells have two features which enables them to do this. First is they can divide themselves time and again to produce new cells, right? Stem, can you just imagine stem? Stem is basically the which lets other things grow, right, in a tree. Like it's not the root, but it's the basically the trunk of the tree, right? So also the second feature is when they divide, they can change into other type of cells. So it's not like stem cells will only become heart cells or lung cells. They can become kidney cells. They can become, let's say, bone marrow cells and so on. So they can change into any cell. Basically, they can become anything. They're like Joker, right? So they can change into anything. So they are special human cells that can develop into different type of cells. They can be brain cells, lung cells, heart cells, anything. Sometimes they can also fix the damaged tissue inside your body. Now, researchers believe that if we can use these stem cells in adults to treat things like paralysis or Alzheimer's disease, so that will be very beneficial. But why it is not happening so much? Because of the way they are obtained. We'll talk about that. So every cell in our body has a specific function. The cell in your eye has a specific function. It cannot be copied, and that cell in your eye cannot be used in your brain, right? But these cells can become anything that you want, right? Stem cells do not have any specific function except to grow. They can grow wherever you put them. So that is one of the most crucial things why research is going on in this. So see, stem cells can create more stem cells. They can also create specialized cells, right? Like blood cells, like this is brain cell and so on. So stem cells, more stem cells, specialized cells. What are the types? There are three types. First is embryonic stem cells. Then there is adult stem cell. So the embryonic ones is found in the embryos, basically the initial stages of a fetus. So point is, obtaining that is it's very sensitive. Basically, the most ethical issue that arises out of it is sometime the child might, basically you're taking somebody else's property without asking them, right? Taking cells from a child's body without asking the child. What if they need it in the future? How are you affecting their growth? So this is the thing. So basically, lot of ethical issues have come up, and people have finally landed on this thing that you can use embryos only which are donated, or basically which are sadly dead. All right, but yes, again, or unusable. I know you might think that if it is dead, then how to use it. So there is some period of it. Again, we don't need to go in depth in this. That will become very technical. So overall, what you need to know is the issue is from embryonic stem cells, because adult stem cells sometimes cannot change into many things. All right, they can turn into more than one type. They are pluripotent. Adult stem cells, they are of two types. One are fully developed cells. They are like very particular, like brain, skin cell, brain cell, bone marrow cell. And in these, there are very small number of stem cells. All right, they will generate only one type of cell basically, right? A stem cell which comes from liver, that will generate only more liver cells. Second type is pluripotent. Now these are adult stem cells which are changed in the lab to be more like embryonic stem cells. So these are the third type. Third type is induced pluripotent. Pluripotent means they can become anything. So that is why due to the issue faced in the embryonic stem cells, now scientists are creating IPS cells, basically induced pluripotent stem cells from adult stem cells, so that this ethical issue does not come up, right? So these are the stem cells that scientists make in the laboratory. These are basically adult stem cells that are converted into pluripotent stem cells, right? Now what is stem cell research? Basically, it's a research to understand stem cells in more ways so that we can grow tissues or organ later on, and we can cure genetic diseases, we can cure diseases that are basically incurable, like Alzheimer's, dementia. Dementia is the early stages of Alzheimer's, and many other things, right? What is the therapy? What is this term, stem cell therapy? Basically, it's a regenerative medicine. What does that mean? That means if there is a tissue which is dead or not working properly, it can be replaced by the use of stem cells. Now, how can you use this? You can use this in organ transplants. Because right now, transplants. As of now, there is a list. In every country, in every hospital, there is a list of organ transplant. So obviously, there are not many donors. So if we can crack this thing, we can crack the therapy, regenerative medicine, we can regenerate the organs which are obviously not available in huge quantity. So there it will help. Now carefully see this animation. This is actually a original video. it is thumping, right? It is doing a motion like the heart. So basically these are the heart cells that were grown from stem cells, and these were created in a petri dish, right? So stem cells created stem hearts, or basically heart cells, which are now pumping in front of you, and we can use them in research. All right. Now we'll come to the important techniques of biotechnology, how things are done and how practically we can use biotechnology. All right. Recombinant DNA technology, genetic engineering. Genetic engineering, we have discussed about rDNA, right? What it is, you know? So what is genetic engineering? Basically, what is the rDNA technology? In this, what happens, like I've told you, in rDNA you can combine DNA strands from two different sources. So in this technology, basically genetic engineering, a molecule, basically in rDNA, a molecule of DNA is modified and there are genes from multiple sources. All right. So if there is if this is one DNA, it can have sources from multiple DNAs, multiple genes, right? So in this, how it is done? In laboratory, scientists use rDNA to extract, or basically to take out some important property, like I've given you example of two bacteria previously, right? And then they add it to another organism. So this is how they create new products which are not naturally available, or new organisms which are not naturally there. All right. One of the benefits which was discovered was DNA is universal. Its design does not change from a person to person. So basically, the best benefit of it is that the DNA can, the structure is same, so it can be simply replaced with another DNA. Getting the point? So this means that you can go cross species also, right? So basically this was done, and a bacteria's pest resistance capacity was put into cotton, like cross species. Let me tell you again. So the structure of DNA or the design of DNA is universal. It is same in every organism on the earth. So the rDNA can be sent from one species to another. That means you can add genes from bacteria to another bacteria, bacteria to another species also, even in humans. Obviously, human thing is not allowed right now, right? So this is very important to understand. See, a simple question can be asked that rDNA technology is used for same species genetic engineering only. All right. Now let's see some examples. The insulin is produced at mass scale by a bacteria which produces the human, basically the human producing insulin cells are taken and put into a bacteria, and that bacteria then produces that insulin. So insulin is produced on mass scale due to rDNA technology only. rDNA, sorry, I don't mean our DNA. I mean, whenever I'm saying rDNA, it means recombinant DNA. All right. It is also used in agriculture. Before rDNA technology, farmers used to crossbreed, right? And they were obviously also trying to make their crops resistant to insects and weeds. But with genetic engineering, scientists can now easily identify what type of bacteria or what type of character in a crop is resistant to a pest. Now they will take out that particular gene which is resistant to pests, and then put it into the crops. Getting the point? Scientists will now identify which gene is resistant to pests or weeds in a crop, let's say, or in a bacteria also. Now they will use that particular thing or that particular gene and put it into a crop, right? Let's say you want to create a let's say, high temperature resistant crop. So you will basically research something which grows in near Rajasthan, and you will take that part of that gene of the plant in Rajasthan and put it into other plant which, let's say, is grown in Delhi, right? So such things are there. Also, one more example that we can take is insect resistance, which is pest only. So how it was actually done, scientists placed the genes from bacteria which was resistant to lot of insects, and then they placed it into the DNA of corn, placed it into cotton. Now the genes they selected produced the protein called BT. This protein was produced, right? So whenever an animal ate it, or whenever a pest ate this crop which has the BT protein, it was lethal for that pest. All right. So scientists did what? They created the rDNA from the genomes of this bacteria which had this protein, and then put it into the crops. So new DNA was created, and because of which the crops are now protected. So whenever the crops grow, like BT cotton, whenever it grows, it produces the BT protein also. Now that BT protein is not harmful to us, but it is harmful to the pests. So please remember that. Next is third example, gene therapy, the application of rDNA. This happens when DNA is introduced into a patient to treat a genetic disease. So we will change the DNA and insert the new DNA into the person. Now that new DNA will have the proper gene that is functioning properly. Obviously, because in the, let's say there is a patient and in them there is some genetic anomaly, like sickle cell anemia is there. Another example can be cystic fibrosis. Again, we don't need to go into detail in that, it's a general studies exam. So we need to just understand how it works. So let's say a person A has some genetic disease, some person B does not. So we can take the gene from B and put it into a DNA and put it into the person A. So the new DNA will have the proper functioning gene which will correct the effect of the faulty gene. Alright, that faulty gene creates a disease in the body. Alright, so for how does it happen? We have to first of all carefully select what is causing that disease, the gene that is causing that disease. That is the first step. Then we combine, basically we do rDNA and then we insert. So this was developed in 1972, but till now there has been limited success because of obviously human ethical issues that you are testing on humans. So people don't really realize the effect and a lot of things. So basically due to ethical issues, these things have been slow. So it can be used for muscular dystrophy, cystic fibrosis, sickle cell anemia. All right, sickle cell anemia was reversed in mice. So that is a very good achievement, but it is yet to be replicated in humans. So coming to the next technique, which is plant tissue culture. Tissue culture is basically growing a single cell from a plant. Okay, and now how will it affect in, or how it is a biotech issue? We'll see that. So basically it's a process in which a plant is grown in laboratory. Now the initial part of the plant is taken and developed in a very controlled environment. Now why do we do this? This is done to, let's say, protect a rare plant which is not able to survive outside. So but it is important for our environment. So we can keep it protected for future generation. Also, such things can be used for testing new crops, which can lead to higher crop yields. So basically for experimentation, plant tissue culture is very important. How biotechnology plays a role in this? Because obviously technology is used in this biological process. And for country like India, this is very important. If we can experiment and create more high yield crop varieties with the help of tissue culture, right? Third technique is transgenics, basically creating genetic modified organisms. Now, this needs a little bit of introduction also. People from ancient times, or rather past, from our very before the technology, people have been altering the genome of plants using traditional breeding. This has been going on already, right? And this traditional breeding, basically what you are doing is humans select the trait they want in an animal, right? And they try to grow that more. Like let's say there is a bull, all right? This happens a lot in horse breeding and bull breeding and dog breeding also. If they see that there is some dog, horse or a bull and they are very healthy and stuff, they have a lot of features that the people want, so they will breed that animal only. All right, so this is basically artificial selection of choosing a particular breed of a dog, let's say. All right, so due to which a lot of different organisms like sweet corn, hairless cat, all these have come up. But this is limited, and this is limited to what? Only natural things. That means you cannot have something which is not there in the organism. But with biotechnology, with the latest technology, you can add something which was not there originally in the animal, like luminance. Basically, there have been experiments where in an animal, they will basically, it's a thing, or pardon me, it's not a published study or anything, it's just a thinking that there can be animals which can glow in the dark, and that is an experiment. It's not been done commercially or anything, right? So just a thought, I'm just saying, it just came into my mind that how to explain you the difference. So natural breeding is having the traits of an animal go on, but only the traits you want. All right, let's say there is a horse which has golden hair, you want more golden horses, so you choose that horse to breed like this. But in artificial, you can insert something which was not there originally in the animal, or even in human, or even in plants, right? So today we can have new genes from one organism and take it to another organism, or even we can take it from one species to another species which are totally unrelated, which can never breed naturally, but with genetic engineering we can do that. All right, so this is very important for pharmaceutical sector and crop, like basic crops, farm animals, soil bacteria. These are the organisms basically who have been subjected to genetic engineering. That is why we have BT cotton, right? All right, so I have explained you this. All right, everything that is written over here has already been explained. So we can now incorporate genes from one species which is unrelated to another species via genetic engineering, by which we can increase the agricultural productivity. All right, also create pharmaceutical substances, new ones. All right. All right, everything is explained. Whatever I said in the previous, like everything is explained, everything is written. All right. So whatever is written, I've already explained. All right, moving on. One thing you need to understand is what is GMO versus transgenic organism. GMO has a modified genome in it. All right, the name is genetically modified organism. So in that organism, gene is modified or genome is modified. In transgenic, it is a GMO, but it carries a gene or part of DNA from another organism. In GMO, normal GMO, you will just change the DNA from same organism. All right, you will change something. But in transgenic, you will have foreign genome. All right, that's it. So basically, transgenic is a type of GMO. Now, last section for today, GM crops in India. Let's quickly see that. So BT cotton is the only crop that is actually allowed in India legally. Now it has two alien genes from the soil bacteria Bacillus thuringiensis that allows crop to develop a protein which is toxic to the pest, right? So whenever a pest eats that cotton, it dies because that is poisonous to that pest, not to humans. So basically we are not eating cotton, but just saying. So this is how research was done. It was identified what is poisonous to pink bollworm, and then that protein producing gene, listen carefully, protein producing gene was taken from the soil bacteria BT, Bacillus thuringiensis. So it was taken from that is why it is called BT cotton, cross species genetic engineering. BT brinjal is also under research. It has a gene which it allows it to resist the pest attacks. All right. So these are the GMs crops in India. BT brinjal is not approved yet. Only cotton is approved. What is the legal position in India? GEAC, Genetic Engineering Appraisal Committee. Again, this was asked in UPSC once. It is an environment thing, but still I'm mentioning here. It is the apex body that allows for commercial release of GM crops. So in 2002, BT cotton was allowed. More than 95% of the country's area is under BT cotton. Now, if you use something which was not approved by GEAC, it can attract 1 lakh rupees penalty and a jail term of 5 years under the EPA, Environmental Protection Act. But sadly, regulation is lacking. So there are many farmers who are using genetically modified crops, but still they are not approved, and this is happening in India. All right. So that is all for this lecture. Thank you everybody. Thank you for listening. Thank you for your patience. If you have any doubts, please let us know. We will be happy to resolve them for you. So this is all. We'll be meeting in the next lecture, in the next part of biotechnology. Till then, take care, do revise, and bye-bye.